So the staff in the proteomics core, we've had some restructuring recently. During COVID, lost a lot of really good people, but we've replaced them with even better people. So Anthony Saviola comes to us from Scripps Research Institute. He's the manager. We still have Monica as the technical manager of the facility, and a recent addition from here in Colorado, David is a PRA. So today I'm going to talk about some of the capabilities of the core, sort of our standard capabilities. The first three, protein identification, is the workhorse, the one that most projects are involved with. Post-translational modification mapping, protein quantification, and then I'm going to talk about some areas where we have a little additional expertise. Compartment resolved tissue proteomics, for example, protein interaction partner studies, so identifying protein complexes, protein interactions, either in vivo, ex vivo. And then two newer areas of service and expertise that we have in the lab, plasma proteomics. And then I'll give an example of a project using the new sensitive instrument, single cell instrument that Angelo mentioned earlier. So the standard workflow and variations of it are represented in this figure. So on the left, we have cells of interest or tissue samples of interest, biofluids also, or even isolated protein complexes. The next step will be extracting proteins and getting those ready for enzymatic digestion. We're going to digest proteins down into peptides, and then we're going to separate those peptides on chromatography, usually 60-minute runs, could be 2-hour runs for better coverage, or on some of the more modern instruments, we'll even use more rapid methods, 5-minute methods for some plasma proteomics or proteomics of less complicated samples. Then in the mass spectrometer, we're going to be measuring the mass of those peptides. We're going to select each peptide for fragmentation. And then from that fragmentation, we can match to a theoretical digest and theoretical fragmentation of all peptides in the proteome and make matches and identifications for peptides. Assemble those peptide identifications up to a protein level report, and that report is going to be what you'll receive from the facility across all samples. So there is a fairly significant amount of data processing that goes into that. And then, of course, on this right-hand side, we have, statistical analysis, further bioinformatic analysis, and that can be achieved through the bioinformatics core through collaborations. And then down here, I've shown some variations on this workflow where you can look at subcellular fractions, nuclear fractions, for example. You can identify protein interactions with affinity pull downs, and I'll talk a little bit about that technology. And then you can look at post-translational modifications from the total proteome that you've measured, or you can enrich for classes of post-translational modifications. We have a range of mass spectrometers from a targeted instrument to do very specific characterization if you know what analytes, which peptides in this case that you're interested in, all the way up to more state-of-the-art instruments. And then I'm not showing the two newest instruments. I'll talk about those at the end of the presentation. So for post-translational modifications, there's an example that we did several years ago. I call it the CPR project. It's the Snow Mastodon project that was up in Snowmass, Colorado. I heard about it driving into work one morning, how they were assembling an international team to do science on these remnants identified in this reservoir that was in Snowmass Village, found during excavation. And the thing that really stood out to me is that they said when they cut into these bone samples, they really smelled like bone, like there was a lot of organic material. And so there had been a lot of work and some controversy about the identification of blood proteins in ancient bone samples. And so I thought if there was a possibility of finding ancient proteins, they would be found in these samples from the Ice Age readily that were preserved so well. And so here I'm showing a glycan analysis on structural collagens. And this is from the extinct bison latifrons. This is from a Menzies sample on the inside of the skull that we were able to isolate. And here are galactosyl glucose and galactosyl modifications on side chains of lysines. And down here you can see what a fragmentation spectra looks like, where you have indications of the disaccharide that are on there, and then sequencing of the backbone so that you can really make very high quality identifications. And what was interesting is that this was a really pretty deep discovery of these PTMs. Like even from modern day samples, it's fairly challenging to find these modifications. So I also found a data set from the baby mammoth found in the ice caps in Siberia, Lyuba, that's approximately 42,000 years old. And from the femur sample, proteomics had been done, and there was very similar patterns of this post-translational modification. In terms of quantification, a lot of projects were going to steer you towards label-free quantification, and this is just comparing, sample A versus sample B, running each, and then for each peptide identification, doing area under the curve measurements for the peptide of interest across the two samples or across as many samples as we have. It works quite well. One important factor, though, is that samples are run at the same time due to variable performance of the instrument over time. We also have expertise in SILAX. This is stable isotope labeling of amino acids in cell culture. So usually carbon-13 labeled amino acids in your media. The cells are going to integrate that into every protein. And now we can use the mass difference of the light form and the heavy form of a peptide to do quantification. And then the third approach, which is probably the second most common, it really has an advantage in that we can multiplex up to 16 samples in the same mass spec run, or tandem mass tag labeling of samples. And so that has a huge advantage that you can use the same amount of instrument time and dig much deeper into the proteome. And then also we have, with that targeted instrument, the capability to do synthetic or labeled internal standards for absolute quantification. So one of the methods that's been developed that we have sort of international recognition for, people come to us, is the ability to characterize tissues very extensively. And so here I'm showing sort of a standard proteomic protocol where you'll extract the proteins, and we'll call this the cellular fraction, usually with a strong detergent. And now here I'm plotting cellular secreted proteins, cytoskeletal proteins, and then a range of classes of extracellular matrix proteins. And you can see then if we use a chaotrope like urea, we're able to extract more of those proteins, and then we use a chemical digestion to get at the insoluble component of the extracellular matrix for much better coverage of tissue samples. So if you do this in, say, the example of the tumor microenvironment, this is a case where we're looking at a KTC model. This is a biomechanical driven model of pancreatic cancer, where you cluster the TGF-beta2 receptor together to create that biomechanical signal. What you can see here is that a range of basement membrane proteins that are important for cellular function in the native tissue are lost in the disease state, and that we have a really significant increase in provisional extracellular matrix in the early disease state, and in the late disease state, some of that has been cleared. And so one of the things that's been very powerful is to now correlate that with proteins in the cellular fraction. So in this case, MARX1 or CD206, a marker of tumor-associated macrophages. And now you can see the extracellular matrix proteins that are correlated with that. So if you were to target this signature, you might do that by targeting your macrophages in this example. One of the really powerful things that we can now do is correlate tissue architectures with the extracellular matrix composition. So this was work done with the late Patricia Keeley, looking at breast cancer. So in invasive ductal carcinoma, she identified a curly and a straight ECM architecture associated with the straight signature associated with more aggressive disease and poor patient outcomes. And so we're able to identify proteins and correlate proteins with those architectural features. And then using immunofluorescence and SHG imaging, second harmonic imaging, we can now correlate whether or not those proteins are part of those structures that are being developed in this tumor microenvironment. Talking about the tumor microenvironment or tissue microenvironments, it wouldn't be complete unless you talk about glycosaminoglycan. We have, in collaboration with Eric Schmidt and Kaori in pulmonary medicine, the capability to characterize glycosaminoglycans. So protein interactions, there's a number of ways to identify protein topology, protein interactions. I'm going to talk about affinity purification, chemical cross-linking, and proximity labeling approaches briefly here. So the traditional method to identify protein interactions is to use an antibody to a protein referred to as the bait protein. You capture that protein, and then using mild lysis conditions, you hopefully retain proteins that interact with it. And in this example, it's shown using an SDS page gel to separate those proteins, isolate protein of interest, and identify by mass spectrometry. These days, usually what we'll do is we'll digest on beads so that we capture all proteins that are present. It's very important then to have a control, like a pre-immune antibody as a mock, so that we can subtract background proteins from specific proteins that might be interacting with that bait protein. And down here, I'm now plotting likelihood of success. These are fairly challenging studies. You have to have a good antibody-antigen pair. And then also there's just a lot of contamination with background proteins. And if your protein of interest is very low level, this is going to be quite challenging because the background proteins will be usually higher than your protein of interest. So then you can make a fusion protein, like a GFP fusion protein, oftentimes people make, and now you have the advantage of using commercial GFP antibodies that are very good. You can also get localization, but sort of a more advanced iteration of this technology is to make a fusion with a TAP tag, a tandem affinity tag. And then one of the features of this technology is that you're going to cleave with a specific protease like TEV. So have a very gentle removal of the bait protein with its interaction partners, recapture it so that you've decreased the background a lot. But really the technology that's transformed these protein-protein interaction studies is the proximity biotinylation, where you take a protein like BurA, which will create a reactive biotin species. It will tag the bait protein and proteins within a small radius of that BurA protein. And now you can use very harsh conditions to isolate any protein that has a biotin tag on it. This has a huge advantage. Now you can do in vivo experiments. You can do experiments of membrane proteins that are very hard to isolate in a way where you've maintained the protein interaction partners. There's two flavors of this technology. There's the BioID versions, and there's modifications of this that are now more rapid. This uses endogenous biotin, and then there's the Apex approaches, which is use a biotin phenol molecule. This has an advantage that if you're in vivo, you don't have to worry about endogenous biotin. So you can give just a pulse of biotin phenol at a given time point. So to get protein topology, a lot of times we use a chemical crosslinker, and now we're going to identify the linear peptides and the crosslink peptides. And if we plot the crosslink peptides on linear representations of proteins, you can see we have intramolecular crosslinks and then intermolecular crosslinks between components. And here you can now use, this is work with Meyer-Churchill's group here, you can now create structural models where you show relationships between these proteins, whether or not you have a structure or not for the different components. And then also in cryo-EM, when you don't have high resolution structures, but you do have high resolution structures of individual components, you can get protein topology from these cross-link constraints. Now, the newer areas that we've been working on are plasma proteomics, and working on this to be a more high throughput assay. And really the sort of breakthrough here was the technology, a higher throughput liquid chromatography system and a mass spectrometer that's been more sensitive and scans faster. And so now we can process samples in a 96-well plate. And here's an example of a plasma proteomics project that we've done with the Trauma Center here and with Denver Health. And so here we've looked at a little over 100 control patients and then a little over 100 trauma patients. And you can see, based on the averages across the 7 to 8 time point sampling of the trauma patients, that we have different kinetics of proteins in the plasma, things that are released during traumatic injury that are going to be dependent on injury type and severity, things that are depleted, a lot of coagulation factors that are either depleted or partially depleted, and then we have all the way down to sort of things that the liver is going to produce during an acute phase response. So if you look at the first hour time point, you can now separate patients in this study based on severity of injury, and you can see there's a drastic increase in primarily intracellular proteins, muscle proteins, tissue damage proteins that occur with higher injury. But what's really valuable is now correlating the proteins, and we've done with metabolomics as well, to clinical parameters to try to identify relationships and get at mechanistic hypotheses that we can go and test. Angelo mentioned this new technology that is being set up in the core. It's a single cell proteomics instrument, really can be used for metabolomics or measurement of any biomolecule. And here's an example of a project with John Rin's group and Federico Ariel, a visiting scholar in his lab up at CU Boulder, where they're using ChIRP-seq, chromatin isolation by RNA purification. So this is a little bit different than ChIP-seq in that you're isolating the RNA, versus in ChIP-seq, you're usually isolating a transcription factor or a protein, or even oftentimes a post-translational modification on histones, and then sequencing the DNA that is co-purified with that, that biomolecule of interest for genomic sequencing. And usually in these assays, proteinase K or another digestive enzyme is going to be used to remove the proteins of interest, because historically, it hasn't been very productive to try to identify those proteins. But with this instrument, we were able to really make a lot of really good identifications. So I'm going to just show you some preliminary data, if my slide will advance. There we go. So this is the 24 samples that were submitted by their group, and in red are the number of unique peptide spectral matches, the number of peptides from the digest of the proteins that came from the experiment, on sort of a really very state-of-the-art instrument, but a few years old, versus this new instrument. And you can see that for a number of samples, there was either no or very low identifications on the new instrument. There was a drastic increase in the number of identifications, up to 4-fold or greater increase. And then here's an example of a protein of interest for them, where a single peptide had been identified on the older instrumentation, and that significantly increased to approximately 12 peptides on this new instrument. And so I'd like to promote this as a pilot project, so people that are doing seq experiments, we can have a financial discount for trying these types of experiments. I think this could be very empowering to be able to take the sequence data that you get and also correlate that with the protein identifications, and in some cases use that for quality control measurements as well. So to find us, we're in this shared mass spectrometry resource that houses the Metabolomics core, Angelo directs, and then the Proteomics core. Anthony's going to be the main contact point. We have a sample submission form that we'd like you to fill out, and we're always happy and actually really suggest that you communicate with Anthony or someone else from the facility to design experiments so that we make sure we come up with the answers that are trying to be addressed with a specific question. And you can find us, here's our web link, but the easiest thing is probably just to do a Google search of Proteomics School of Medicine at CU. Yeah, so I'll leave it there. Thank you for your time. If there's any questions, we'd be happy to answer those now.
Thank you both. I don't see any questions in the chat either. We appreciate you presenting today.
Right. Thanks, everybody.
Wait, I have a quick question.
Yeah.
This is Chris. Very nice presentation. I learned a lot from this and all the potential, opportunities. I have a grant that's due soon, and we'll be collecting lots of samples, blood samples. What would be the best way to store this for future use, pending extra funding for, the different types of samples that would be valuable? How would we store it? I know plasma sera, but if we wanted to do anything with whole blood, how would you store that, or can you? This is for biomarker discovery.
Yeah.
Biased biomarker discovery.
So for proteomics, it's just going to be -80 storage.
Of whole blood, you could just store the whole blood?
Yeah, of the whole blood. Of course, once you've frozen whole blood, you're not going to be able to separate the cellular fraction from the fluid phase anymore. So if that's important, then you definitely want to do that before you do your freeze cycle.
But we should save sera, plasma, and just freeze whole blood.
Yeah, Angelo, unless you have another recommendation.
Well, yeah, if the study is for whole blood, that doesn't really matter. But if you want to collect, plasma and platelet-rich plasma and red cell pellets, I would recommend like EDTA tube collection. Hence it works best, at least for metabolomics, and separate in different tubes and store -80. We have had samples that have been in the freezer for up to 7 years, and we still got something reasonable out of it. There are biomarkers, especially some specific oxylipins, that are markers of chemical oxidation in the freezer versus enzymatic activity. So we can use that for specific characterization of potential aging of the sample in the freezer. That said, my understanding is that will be within a 1 to 2 year time frame, so it should be really easy to just process everything at once.
And any species, right?
Any species. For metabolomics, of course, any species. For proteomics, you need to share the species in advance for searches against the appropriate genome.
All right. Thanks.
Yeah, for proteomics, you either need a genome or some good RNA-seq data to assemble a protein FASTA database that would be used for identification.
Yeah. Okay. Thanks.
It looks like Kevin has a question. Yeah.
It's Cole, actually. Well, yeah, I'm with Kevin.
Okay.
do you do for the interactors, do you run that apex reaction, or do you usually have the labs that are working with the protein just look into it and run it? Yeah,
in my lab, we have the plasmids to do that work, but that's going to be something that will be done by a user of the facility. It's a fair amount of work to do the cloning, of course, to make the fusion protein with the apex. But we definitely consult and help design experiments, but that will be something that will be done by the user.
Okay. Yeah, I was just wondering, I thought that was interesting to see that technology.
Yeah.
That was all.
Great. Thanks.
Thanks, everybody.
Yeah. Thank you. Have a nice day.